Hello, my name is Damian Wrigley, and I am the National Coordinator of the Australian Seed Bank Partnership. First off, I would like to welcome Mr. Richie Allan, who will be giving the welcome to country.
Hi, my name is Richie Allan, and I am a Ngunnawal knowledge holder of the Ngunnawal Nation. First, I extend my respect to Aunty Violet, a Ngunnawal elder who has passed the traditional stories and knowledge to me. I also extend that respect to Uncle Brucey. I am here to perform a welcome to country. I would like to start by paying my respects to the Ngunnawal ancestors. It's the sacred footprints that they left behind for us to follow that we have survived for so long. It's important in this country we call Australia that we protect those footprints. There are many stories, there are many dreamings in these footprints, and it is the future of this land we call Australia. I would also like to pay my respects to the Ngunnawal elders and thank them for the keeping of the land and the passing of the knowledge that they do to the next generation. I also extend that respect to other brothers and sisters that are here today, as well as other non-Indigenous cultures that are online today. A welcome to country is an ancient practice that has been alive for over 100,000 years, and it will still be around for over another 100,000 years more as we pass the ancient practice down from generation to generation. This practice ensured cultural safety while on another country. It ensured respect. It ensured the taking of everything when you left that Aboriginal country. This ensured the dreaming was never interrupted. In the words of Aunty Violet, Yan Yalamandi Nanadara, Yaramulan Nana Gur, Nangi Yaramulan Nana Migiri. In English, hello and welcome from Ngunnawal country. Dreaming from the Ngunnawal people's country's heart. Seeing the dreaming through Ngunnawal people's eyes. Yara, thank you very much.
Peoples, and pay my respects to their elders, past, present, and emerging. I also extend this acknowledgement and pay my respects to all First Nations peoples that are joining us this week. So it is good morning, good afternoon, and good evening to our special guests, keynotes, committee members and delegates, and welcome to the Australasian Seed Science Conference 2021. It's been a long time coming. Following last year's postponed conference, we have worked hard to bring you a program that does justice to what we set out to achieve last year, taking into consideration the massive global changes since, and I hope we have managed to do so. I think we have an exciting and absolutely packed program this week, and I would like to thank the organising and scientific committees for their significant contributions over the past few years. In particular, these leading women scientists, our chairs, Dr. Sal Norton and Dr. Cathy Offord, and the manager of our host seed bank, Dr. Lydia Guja. I would also like to acknowledge our partners and our sponsors. In particular, the Council of Heads of Australian Botanic Gardens for supporting the Australian Seed Bank Partnership to run this meeting once again. As you will soon hear from Dr. Cathy Offord and Dr. Lydia Guja, this year's conference follows on from our previous meeting, the National Seed Science Forum in 2016, which highlighted the diversity and complexity of seed science that delegates were undertaking at the time, as well as identifying future priorities. Seed science is fundamental to ensuring the survival of our native flora. Seeds are also fundamental to our own existence. They become the food, fibre, medicines and shelter that all life on Earth relies on. They inform our sense of identity and belonging, helping to share the diversity of cultures that we share with each other today. Our survival is intrinsically linked to the seeds we collectively seek to understand and conserve. Your work, and the work of countless more seed scientists around the world, is an inspiration to many and contributes to the foundations on which our future survival will depend. I implore you to continue sharing your knowledge, respecting and acknowledging the cultural significance to First Nations peoples. Continue sharing with each other and extending your networks to ensure seed science continues to be understood and valued for how vitally important it is. And lastly, I would like to thank the Australian National Botanic Gardens for hosting this year's conference as a virtual event. As you will see next, the Gardens is, at its heart, a scientific institution, with seed conservation a key component of its current and future efforts to support ex situ conservation of the Australian native flora. So I would now like to welcome Dr. Judy West, Executive Director of the Australian National Botanic Gardens and our host for this year's conference, who will welcome you today.
Hi there. I want to welcome all of you to this virtual event hosted by the Australian National Botanic Gardens. Now, I can't see you all, but I know many people are attending, so welcome. I'm Dr. Judy West, from the Executive Director of the Australian National Botanic Gardens. I'd firstly like to acknowledge and thank the Ngunnawal Elder for welcome to country, and also to mention the First Nations peoples on the lands from which many of you are participating today. So it's either good morning, good afternoon or good evening, depending on the time zone you're in, and welcome anyway. So we were obviously very keen and looking forward to hosting this meeting last year in person in 2020, which would have been part of our 50th birthday celebrations, but that wasn't to be. The Australian National Botanic Gardens has concentrated on Australian native plants since its beginnings, and has built up the collection, a scientific collection that is much more than a garden. And these gardens, together with our joint venture partners in CSIRO, have a long and rich history in the botanical sciences. And over the past 10 years, our National Seed Bank and its role in the Australian Seed Bank Partnership and its research program has developed and grown so much that we're at the stage now of planning a new National Seed Bank, which is an exciting venture. This is complemented by the fact that the Australian National Botanic Gardens hosts the National Coordinator of the Australian Seed Bank Partnership, which is a position that supports and leads the national strategic approach to seed conservation across the country. We wanted to celebrate, obviously by holding the meeting here, we wanted to celebrate our leadership in Australasian seed conservation, and to show the commitment and passion that we have for seed sciences. And I hope you'll get a feel for that during the week. It's a shame and disappointing that we aren't able to host you personally, but I think there's a couple of upsides to that, and one is that by having it virtually means that some of you have been able to attend who may not have been able to travel to Canberra. And on a point of positive point, since we can't host you personally, we have prepared a virtual tour of the gardens to show you some glimpses of some of the activities and the gardens themselves. And so where we would have welcomed you, but hopefully this will give you a bit of back of house indication of the botanical sciences that are happening in the Australian National Botanic Gardens. So that's all from me, but I hope you do enjoy the conference. I find it fulfilling, and then obviously join this. I hope you enjoy this small video. Thank you.
The Australian National Botanic Gardens is home to the world's most comprehensive display of living Australian plants. We're growing over 4,000 different species here, almost 1/5 of the Australian flora. In fact, it's the only place in Australia you can experience that level of diversity of living plants in any single location. While visitors can enjoy these beautiful displays of native plants and gardens, behind the scenes is a vast array of research, discovery, and conservation work that plays a vital role in conserving our native flora. And with an increasing number of native plant species under threat, this work has never been more important. While all botanic gardens are established in part to enable plant research, the Australian National Botanic Gardens is an internationally renowned scientific institution. Here at Black Mountain, specialist teams based at the gardens and next door at the CSIRO work together to research and document Australia's biodiversity, to study and maintain plant populations, conserve rare and threatened species, and provide specimens and information for scientists across Australia and around the world. Working in partnership with plant conservation organisations, land managers and other botanic gardens, they provide expertise and a national focus for research and conservation, working on projects across Australia. Through the National Seed Bank, the Gardens has a critical conservation role, collecting and providing long-term storage of Australian plant species, particularly those that are rare and threatened. It also has a national and international reputation for developing new knowledge about seed biology and ecology to enable the conservation of threatened Australian plants. And with an estimated 70% of germination cues for Australian flora still not known, the research done here with partners and universities is vital for protecting and preserving threatened species. The Gardens Nursery team works closely with the National Seed Bank and partner organisations on research, using the stored collections and germination protocols to test cultivation and seed orcharding techniques. And where seeds are in limited supply or simply not available, they work with living samples of leaves and shoots collected by garden staff and volunteers in the field, propagating them in a range of purpose-built facilities here on site. As experts in growing plants that are poorly known and highly threatened, they develop innovative cultivation techniques. And to ensure that each plant's viability and genetic diversity can be tracked and studied, strict hygiene and data protocols are followed at every stage of their work. Many of the plants cultivated in the nursery from seeds and cuttings are provided to the living collection team to secure the species here in the grounds. This provides an invaluable resource for researchers who can study them here rather than visiting ecologically sensitive sites. Like putting together the missing pieces of a jigsaw puzzle, this process of germinating, cultivating and growing threatened plants here on site builds understanding about how to care for them here in the gardens, how to care for them in the wild, and ultimately how to secure their future. In some cases, plants propagated in the nursery are returned to their natural habitat as part of replanting programs to renew wild populations. This is particularly important as the number of species remaining in the wild may be so small that just one bushfire or extreme weather event could lead to their extinction. Importantly, every seed or cutting that's collected can be traced to its original source, the plant or population it came from in the wild, and its precise location, through carefully preserved specimens stored in the Australian National Herbarium. This collection of over 1.2 million specimens is a record of plant biodiversity in Australia, spanning 2 centuries. ranging from specimens collected by Joseph Banks and Daniel Solander to those collected each week by gardens and herbarium staff and volunteers. The Australian National Herbarium is a valuable data source, mapping the distribution of Australian plants over time. And it ensures that scientists and researchers can be confident of the origin and the identity of plants in the collection, providing the foundation for their work. These close links between the Australian National Herbarium, the National Seed Bank, and the Gardens, Nursery and Living Collections teams puts the Australian National Botanic Gardens in a unique position to help study, conserve and secure Australia's threatened plant species, now and into the future.
Thank you, Judy and the team at the Australian National Botanic Gardens. I would now like to introduce Dr. Cathy Offord from the Australian Institute of Botanical Science at the Australian Plant Bank at Mount Annan, and Dr. Lydia Guja from the National Seed Bank at the Australian National Botanic Gardens, who will be providing the research front synthesis of the National Seed Science Forum 2016.
Cathy Offord. Lydia, Guja and I are happy to be welcoming you on behalf of the Scientific Committee and Organising Committee and speaking with you about the research that has led to this conference. Seed science is fundamental to many fields of endeavour, from agricultural production to restoration and conservation of threatened species and communities. This is especially the case in Australia and beyond, and we have seen hugely increased involvement and outputs from seed scientists across many sectors over the last 3 decades. Reflecting the rise in extent and diversity of seed science in Australia, in March 2016, the inaugural National Seed Science Forum was held at the Australian Plant Bank in New South Wales. The forum was attended to capacity by over 140 delegates from 9 countries, from government, academia, non-government organisations, industry and volunteer groups who came together to discuss seeds within the 4 central themes of dormancy and germination, establishment and management, seed storage, conservation and utilisation, and seed ecology. A key purpose of the forum was to bring people working with seeds across different sectors together to share the latest research and ideas, discuss contemporary issues, and form new collaborations to advance future conservation, agricultural and restoration efforts across the country. During the forum, there were 67 oral presentations and 18 posters displayed, and a panel discussing the Indigenous use of seeds. The range of topics was diverse, covering rare and invasive species management, the use of seeds in restoration and direct seeding, as well as seed banking of crop wild relatives, recalcitrant species, and rainforest taxa. Other themes covered included seed germination ecology and issues of germination as they relate to ex situ seed bank management, as well as the potential implications of climate change on in situ germination and seed banking. The topics were summarised in a synthesis paper that was the introductory paper in a special issue of the Australian Journal of Botany, dedicated to seed science. The 2016 Forum program highlighted that a lot of research was occurring on the topic of seed conservation and genetic resources. Seed banking is a powerful tool for conservation, and seed banks provide a safety net for securing genetic, species and community level diversity of plants. But it is the sheer diversity of Australian flora that constantly tests the ingenuity of those engaged in seed conservation. With biological diversity comes a diversity in seeds' responses to the standardised environmental conditions employed in seed banking. Defining and predicting seed storage behaviour is clearly an area of need in conservation and agriculture, particularly as seed collections increase in size and curation activities become increasingly complex. Another common theme at the forum was seeds in species translocation and landscape restoration. Seed scientists have a major role to play in supporting habitat restoration through the sourcing and provision of seeds and through developing expertise in seed-based propagation. Contemporary restoration programs aim to restore biodiverse plant communities at the landscape scale, meaning that the return to tens of hundreds of species will be required in many ecosystems, and there is increasing focus on enhancing the diversity of understory in restoration programs. Thus, determining seed dormancy and germination traits, historically perhaps the primary area of research focus in Australia, remains a fundamental need. Other areas such as seed ecology and seeds and climate change research demonstrated the emerging recognition that seed regeneration traits can also be employed to study the effects of changing climates on species distributions and vulnerabilities. We also saw some studies applying molecular biology to seed biology questions, and we observed increases in collaborative work to consolidate, manage and share data and share knowledge between groups. Many of these themes appear to be continuing into this upcoming 2021 program, and we look forward to hearing the developments that have occurred in these areas over the past 5 years.
Thank you for the excellent summary of the previous forum, Cathy. Since the last forum, we've seen new challenges emerge or intensify, such as widespread and rapid climate change, megafires across 8 million hectares of southeastern Australia, and widespread drought in many of our agricultural regions. All of this creating an urgency and a demand for seed science. That need for seed science is what led to the overarching theme for this 2021 conference, linking seeds with needs. Building on the themes and the emerging research that came from the 2016 forum, we have 4 themes for this conference, which you will notice form the structure of the sessions. Also informed by the previous forum, and the surveys undertaken afterwards, we expanded into a full conference format and to be more international in scope, encompassing the Australasian region and beyond. Through this meeting, we hope that we will together develop a roadmap for seed science and seed use in Australasia. So on behalf of the conference hosts, chairs, scientific committee and organising committee, welcome to all of you. We really do look forward to discovering the future research directions that will emerge from this 2021 meeting.
Thank you, Dr. Offord and Dr. Guja. There was an excellent synthesis of what was several days of inspiring seed science, and I think it really helps to set the scene for this year's conference. I would now like to introduce Mr. Costa Georgiardis, landscape architect and host of Gardening Australia on the ABC. Costa is a strong supporter of plant and seed conservation, and we are excited to have you here to officially open the conference for us, Costa. Over to you. Hi everyone. Unfortunately, we've had some issues with the video for Costa, who will be opening the conference for us. So while we figure those out, what we'll do is we'll go to our chair of the next session, Professor Adrienne Nicotra, who is introducing our next theme, seed biology and evolutionary ecology. Adrienne comes from the School of Research Biology at the Australian National University. Thank you, Adrienne.
Thanks, Damian. A real pleasure to be here today. And I just wanted to thank Ricky for that welcome to country, and to add my acknowledgement to the traditional owners of the country on which we all meet. For me, it is the Ngunnawal people. It's a real pleasure to introduce Xizhong Chen as our first keynote speaker. Xizhong was here last year when we had to cancel and she had to return home. So it's really exciting to actually be doing this. Xizhong is speaking to us from the Millennium Seed Bank at the Royal Botanic Gardens in Kew. She completed her PhD though here in Australia at the University of New South Wales before moving on to a postdoc in Israel and she's going to be giving us a fascinating talk on allometrics and scaling. And I will waste no more of her time, but pass over to you, Sijong.
Hi, everyone. Hi. Good morning, everyone. Can you see my slides? Is it okay now? Because
It's all good, Sijong. Go for it.
yeah, all good. Okay. Thank you so much. Good morning, although it's midnight in Greenwich Mean Time now for me. Thank you for having me. I want to thank the organizers for inviting me and providing me this opportunity. Aside, I'd like to talk about biological scaling in seed functional components. Because I'm an ecologist, I refer to the broad definition of seed as diaspora, depending on the context. So obviously size matters, from the smallest microbes to the largest mammals and plants. Organisms spans a mass range of over 21 orders of magnitude. This vast range even exceeds the range of the Earth's mass relative to the galaxy mass. Ranging from mouse to elephant, the body gets bigger in general, hearts beat more slowly, brains get bigger, bones get proportionally shorter and thinner, and lifespans lengthen. Many of the most fundamental biological processes manifest an extraordinary simplicity when viewed as a function of size. Allometry, also called biological scaling, is the study of size and shape of organisms, their organs, metabolic rates, life history traits, and other measurable quantities. Overall, it describes how traits and processes scale with one another and applies for almost any co-varying biological measures, such as within and or among biological traits, physiological traits, or ecological traits. The study of allometry allows us to link form to function, identify patterns and differences, and simplify complex biological processes. The relationship between the two measures is often expressed as a power law equation. We can convert the equation into a linear relationship by plotting these two measures against one another with log-log axis. The slope of the line is known as the allometric coefficient. If A is equal to 1, we call it asymmetry. If larger than 1, we call it positive allometry. If A is smaller than 1, allometry. Biological scaling is a general law in nature and has been found in several plant life history traits, both across and within organs. Just to give some examples, shoot growth rate scales with root growth rates. Size scales with female leaf size, so do their stem sizes. Within the same leaf, laminar size scales with petiole mass, laminar petiole mass. So these are all with biomass allocation patterns within vegetative organs. Trait scaling has been much less studied in reproductive organs. Meanwhile, less attention has been given to the relationship between within organ components. Seed masses range over 12 orders of magnitude across present-day species, from the dust-like seeds of orchids to double coconut. A diaspora has been usually treated as an integrated unit in its ecological and evolutionary characteristics, despite the fact it consists of several functional components: disposal components, defense component, and growth component. We actually did not know much about the trade-offs between these components. By partition biomass allocation, we aim to understand the variation of seed functional components. We conducted 3. The first one is an intraspecific study. This work was done while I was doing my postdoc with Dr. Giladi. We use growth. It's a wind-dispersed annual herb in the Asteraceae family and widespread in the Eastern Mediterranean region. We sampled seeds across 3 populations in the southern Judean lowlands in Israel. We weigh each individual seed for the 3 components. The plumed pappus constitutes the disposal component. The seed coat and fruit wall are fused and function as the defense component that impedes seed predators' approach to seed reserve. Seed reserve, also known as seed kernel, consists of the nutritive tissue involved in germination, including embryo and endosperm. Overall, the masses of seed components were highly correlated with each other, with high and goodness of fit. There is an asymmetric relationship between purpose mass and seed coat mass, seed level and the plant level. That means relative mass allocation between dispersal and defense components holds consistent. There is a negative allometry between seed reserve mass and seed coat mass at both the seed level and the plant level. Defense component scales at a lower rate than seed reserve. The three populations we sampled have similar mean in their habitat, but their annual precipitation from 450 millimeters north to 300 millimeters in the south over a short distance of only 30 kilometers. So to south, purpose ratio increases, seed coat ratio increases, ratio decreases. These variations suggest seeds in drier sites may be more likely to be dispersed further and escape their maternal sites. So the second work is an interspecific study for defense component versus growth component. This work was mainly done with my collaborator, Professor Bowang, and our student, Lame, also with help with my Q colleague, Dr. John Dickey. Again, to remind you, we divide the seed into seed coat and seed reserve. Seed coat provides a barrier against external disturbances and acts as a channel for transmitting environmental cues. Seed reserve is the embryo-con endosperm fraction, compressing available nutritive resources. Seed coat and seed reserve originate in different parts of the ovule, play different roles in plant life strategies, and are subject to different selection pressure. Their absolute masses and relative proportions influence a shoot of ecological processes. We compared a global dataset of seed coat mass and seed reserve mass from literature synthesis and our own measurements. In total, our full dataset comprises 940 species from a wide range of families and genera. The allocation in seed components varies substantially across species, with no particular pattern at taxonomic levels. Seed coat mass and seed reserve mass each spans 7 orders of magnitude, so seed coat ratio ranges widely from 0.01 to 0.99. These are the seeds of some species you may be familiar with: the tropical fruit Ramadan, which I really like, and the double coconut and Queensland nut. So we find a weak but significantly negative along seed reserve mass and seed coat mass, which results in a negative relationship between seed mass and seed coat ratio. This finding suggests smaller seeds invest proportionally more biomass in protective tissues than do larger seeds. The absolute amount of seed coat in large seeds may reach a threshold that is sufficient to deter most predators in surrounding environments. We estimated ancestral states for evolutionary rates for seed coat mass and seed reserve using a multiple variance Brownian motion model. Rate allometry along single branches of phylogeny using an analytical framework quantify possible co-evolutionary scenarios. Was first used in animal evolution to quantify the relationship of possible evolutionary changes in 2 traits, like in this case is the body mass versus the brain mass. So this approach divides the relationship into 4 quadrants to describe which trait increases or decreases more or less than the other, reflecting all possible evolutionary scenarios of change between traits. So in our case, the evolutionary rates is the evolutionary rates in seed coat mass and seed reserve mass. So across all ancestral descendant pairs, seed coat rates against seed reserve rate, consistent with an overall trend of mass increasing. Both rates in most lineages. We also collect key life history traits. We find desiccation response is the main biotic driver of seed mass allocation. Seed coat ratio of desiccation tolerant seeds is on average threefold greater than desiccation sensitive seeds. The scaling relationship of seed coat mass and reserve mass is significant within either DS or DT seeds, but shows a significant difference across the two groups. This difference lies in the intercept, but not in the slope. That is, the mass allometry of seed coat and seed reserve is independent of desiccation response, while desiccation tolerant seeds allocate proportionally more biomass in seed reserve than do desiccation sensitive seeds. We also determine if the scaling trend of coat versus reserve in a trait coevolution follows the same pattern across response. We used all rates different model to extract the best condition for ancestral state for each node. Again, both rates are positive in most lineages. A minority of DT lineages show decreases in coat mass versus reserve mass. Almost all DS fall in the evolutionary scenario where reserve mass increases relatively more than coat mass. We calculate orthogonal deviation from asymmetry for each branch. The value between DS and DT nodes are significantly different. That means desiccation sensitive lineages and desiccation tolerant lineages show different rate allometric patterns and magnitudes of deviation from asymmetry. So thirdly, we conducted an interspecific study for versus another disposal component. Seed disposal is a complex process be transported in one or more sequential phases, potentially involving several disposal agents. Many species are associated with a single phase disposal syndrome known as monochory. So here we study diplochory. It is the case where species dispose their seeds in two separate phases, usually associated with different disposal structures and presenting different types of adaptations to different disposal agents. Compared to any single phase disposal syndrome, diplochory is believed to increase this seed disposal effectiveness, reduce seed predation, and yield a double seed bank. Phase 1 disposal scatters seeds away from parent plants and usually in lower density. Phase 2 disposal moves seeds to a further distance and to more suitable habitats for recruitment. Several types of diplochory have been identified. One common type is where seeds are first dispersed by explosive ejection and then further dispersed by ant. The Euphorbiaceae family is one of the major taxa with this type of diplochory. For the ballistic disposal phase, the dehydrated fruit coat mechanically propels seeds in various directions. For the ant disposal phase, the lipid-rich elasms function as a nutritional reward to facilitate seed removal by ants. The relative importance of the two phases varies among species, and diplochory may favor one phase over the other. We sampled 91 Euphorbiaceae species from botanical gardens, nature area, and 6 herbaria. The species are from 21 countries. So for these 91 species, there are 68 diplochory species and 23 monochory species where seeds have for ant disposal, like for example, like this one. Whenever possible, we measured the dry mass of seed coats elysomes, and seeds. We find species having a larger component for ballistic disposal also tend to have a larger component for ant disposal. Fruit coat mass scales against seed mass positively, lower rate than does elysum mass. So that means species with larger fruit coats invest proportionally more in than species with smaller fruit coats. So this result reflects a higher reliance on the ant disposal phase. Influencing the relationship between these two disposal investments, the positive allometry between fruit coat mass and elysum mass persists regardless of seed size. The findings overturn some of our traditional assumptions and advance our understanding of diplochory. I want to highlight the story behind this paper, which is a brilliant collaboration between Iranian and Israeli researchers, together with researchers from several countries, Czechia, Spain, and New Zealand. And I'm a Chinese, and I was doing my postdoc in Israel at that time. So finally, take-home message. We find masses of seed functional components exhibit substantial variation and are tightly correlated with each other in a scaling manner. We overall our quantification of the within decibel biomass allocation across multiple ecological levels improves the understanding of plant reproductive strategies. Disposal investment and defense investment are similarly important. Seed coat ratio is negatively correlated with seed mass. Evolutionary rates in reserve mass significantly outpace those in coat mass. Relative investment in ant disposal increases more compared to that in plastic disposal. So finally, I want to thank organizations and funders that provide me my fellowship and research grants. I want to thank Australia Seed Bank Partnership and Friends of AMBG for sponsoring and supporting me the trip last year and finally the conference this time. And thank you very much for listening. Here are my contacts if you'd like to get in.
Thanks so much, Sejong, for a lovely talk. And thanks also for opening our very international conference with a talk that demonstrates how broadly seed science is a collaborative effort across the globe. And I'm really sorry that you have to be up at midnight to give this talk.
Okay.
I might kick off with a question of my own. That's the chair's prerogative while we wait for people to figure out how to use the Q&A. I think, in allometrics, it's hard to get your head around, but it's really fascinating stuff. And the seed coat to seed mass element really interests me because obviously that's your sort of surface area to volume question, right? And we wouldn't expect them to scale perfectly the same. Otherwise, the embryo would get relatively smaller in a sense. Did you contemplate like any null models? Is there a way to sort of have a null model for this sort of scaling? And what do you think the optimal, how would you figure out what the optimal seed coat to seed mass scaling relationship would be?
Yeah, the null model is the asymmetry. So theoretically, we think if one part of the organism increased, the other part should increase in the same proportion. So the null model is the asymmetry. So every time we compare the coefficient with asymmetry, if it's lower than asymmetry, then if it's lower or higher than the asymmetry, then one part or the other should increase more or less than the other one. Regarding the optimal, I don't think there's a finite answer for that because it's also that's also the That's also the reason for doing such research, because it sounds like a common science, like when if our body get bigger, our heads get bigger, brain get bigger, but how much is that? How we haven't quantified it yet. And this such allometric topic has been very well studied in animal world, but not really that much in plant world, and even much less in seed science.
Yeah, absolutely. There's a couple questions coming in now. Sean Walsh is asking, you told us about desiccation tolerance and how that affected the allometrics. Did you look at dormancy type and how that affected these sorts of allometric scaling relationships?
Yeah, we indeed checked dormancy. It's we're still doing this. In basically, yes, there's difference in the mass scaling between across different dormancy types, but the effect of dormancy on this scaling is not that much. So the number one factor among all the biotic factors we quantified, the number one factor is desiccation response.
Yeah, thank you. Another question that's come in from Bradley Desmond is if you're going to scale the study up, a different sort of scaling, what would your priorities be for future study in this space?
That is a good question and also a hard one. Generally, we're still working on this aspect. We're trying to quantify whether the biological, also when we speak biological scaling within the ASSC, as you see the examples, that means different things regarding what ecological aspects we're quantifying. So, for example, whether it is seed coat mass versus seed reserve mass, and probably in future we can also focus on the embryo size versus the rest of the seeds. That's also important aspects and has been very little touched. But I think I'm quite interested in what kind of biotic and abiotic factors that are affecting the evolutionary and biological patterns of the mass scaling seeds. So what is the most important environment factors? Both biotic environment, biotic environment and abiotic environment. What is the number one factors? But that's really a hard question. I'm still also thinking about it.
As you said, it's kind of remarkable that this area hasn't really gotten much attention, and now that you've started going this way, there's obviously so many things you could look at. Catherine Whitehouse was just asking, what about seed longevity? Have you started looking into, we would anticipate, right, that seed coat mass would scale with seed longevity potentially, but what about that allometric relationship and longevity?
That is a really good question. I haven't touched for these aspects, but because also I think for seed longevity is how we quantify seed longevity. This may be, if we use different methods, this can have different patterns related with what we may find for the result. It's probably also I haven't really do much thing about this, but that's indeed an interesting aspect in the future.
You've got me thinking now, because you showed the picture of the elephant in the first place, and the mouse. I'm thinking about skin thickness versus seed coat thickness, and wondering if any animal ecologists have looked at the allometric of skin thickness. But we might have to leave that to them, I suspect.
Also speaking about the seed longevity, what what I was thinking is here we quantify the mass allometry. There's also, if we consider the impermeability of seed coat, that could be, we may look into more details about, because it's not mass is just absolute mass, so it doesn't probably necessarily represent the structure, physiology aspects. So, so things may be kind of complicated if we think about how this mass being constructed at the molecular or cell levels.
Yeah, absolutely. And I suppose the other thing you could start doing is thinking about the structural characteristics and the thickness and the strength of the seed coat. I suspect we could keep going and ask you more and more questions. And I really encourage people. I understand there's been a little bit of trouble with the Q&A, Xizhong. Not everyone's questions in, but this conference is just beginning, and we've all got each other, you know, on the stage for the whole coming week. So please get into the hub on the portal, and you guys can ask Xizhong questions to your heart's content as the day progresses, or maybe over the next few days, because Xizhong might actually go to sleep, I suppose. Thank you so much for a fantastic talk and for getting us started with some really interesting questions to grapple with.
Thank you so much.
I'll pass it over to Damian, I believe, who's going to try again.
Thanks, Adrienne, and thanks for bearing with us, everyone. We know there's a few technical issues going on in the background, and we're working hard to try and fix them. But right now, I'd like to reintroduce Costa Georgiardis, landscape architect and host of Gardening Australia on the ASSC. Hopefully, we'll be able to bring his video with sound this time. Thanks, Costa.
Hello, testing. Can you hear me, everyone? I think you can. And I am super excited to be here to prepare the launch of the conference. But first, I'd like to say a big thank you to Damian Wrigley, Judy West, and the organising and science committees for pushing on and making the conference happen this year. What a great result, and how important that is to be able to share all of the great work you're doing and to make valuable connections. To start things, I'd like to acknowledge that I'm coming to you from my home here in Sydney on Gadigal and Bidigal land of the Eora. And I acknowledge elders past, present and emerging. When we talk about caring for country in the context of First Nations history, seeds and seed science always has and always will be an integrated part of their cultural land management practices. Seeds are a very special language that can break down barriers and grow a future of understanding together. It is a real honour to be here as part of the 2021 Australasian Seed Science Conference. These really are challenging times. And it's very easy, as you would know and as you would see in your communities, it's super easy to be overwhelmed with anxiety about the future. You don't need to look far to see that our conservation and agricultural sectors are being challenged on so many levels, whether it be fires, floods, drought, new pests and diseases. Biosecurity is everyone's business. So it can be so overwhelmingly negative, but wonderful work is being done. Priceless advocacy and campaigning is spreading the word. Look at the new National Seed Bank at the Australian National Botanic Garden, where we all would have been had this happened in person. Along with the new conservatory, 2 projects underway and very soon to be coming out of the ground. And then we have the new plant bank and herbarium at the Australian Botanic Gardens in Mount Annan. These are vital fulcrums to help raise seed science to the heights and reach it deserves nationally and internationally. We're all part of this drive for changing priorities. Now, seeds, they represent hope, change, and the ability to transform. You know that this world is built on plants. Plants provide food, fibre, medicine, and shelter. They are the very backbone of our landscapes, our human settlements, and our communities. And the understanding of our plant kingdom holds the key. Scientific rigour is needed now more than ever. Why? Because we need science to solve these challenges. They're moving faster than at any other time in history. And we need the science. We need to work together in strong partnerships that cross sectors and geographic borders. But when you think about partnerships, they're based on communication, which is cued by signals. Our ability to communicate, to read signals, to feel the temperature and the pulse, and to connect with appropriate tone and context. Our capacity to work intergenerationally is the key. To communicate across multiple platforms, multiple generations, cross-culturally. What a challenge. And guess what? Seeds are critical in addressing these challenges. Seed science is a growing field across both agriculture and environmental conversation. And many people are dedicating their entire careers to solving some of these most challenging germination and dormancy issues across many families, genera, and species. What a workplace, what an industry, what a lifestyle, what an environment in which to grow change, to create outcomes that benefit the collective long after we have become the compost to feed the next generation of seeds. Now is the time to collaborate, connect, and most of all, amplify the merits of seeds to our collective future. Science communication is our business. Seed science communication needs to be an exercise that we all exhibit every day. Bring people into the conversation as individuals, as friends, as family, as colleagues, and of course, community. From land care to greening Australia, bush heritage to conservation volunteers, citizen science to iNaturalists, Questa Game to Frog ID, preschools to high schools, universities to aged care homes. Seeds are the intergenerational glue that we have the opportunity to be pasting connections wherever, whenever, and with whoever we can connect with. Yes, all 350 of you delegates who are here for the conference. You are the glue. You are the amplifiers. I'd like to give a big welcome to many of the Australian participants who are from seed banks in Begans member gardens or are members of the Australian Network for Plant Conservation. You are I'm taking a breath because, you know, this is it. This is the moment. You're the group. You are the group. We need you to connect. And remember to stay in touch after the conference. You're the 2021 cohort. We need this network to grow and grow. So it gives me great pleasure on behalf of the organising committees to declare the Australasian Seed Science Conference 2021 open. Go forth, everyone. Sow the seeds for a connected future.
Thanks so much, Costa, for an enthusiastic opening for our conference. And you are so completely right. This is the group that we need to get talking and networking and working together. And the virtual conference, as Judy said, is in a sense an opportunity for us to do that across the globe without having to worry about the limits to travel. Just a quick bit of housekeeping. I've been asked to remind you all that Chrome is the preferred software to be using here if the portal is misbehaving for you at all. We'll now move straight into our first series of contributed talks, and we've got all sorts of really cool work on functional traits in response to water stress, to snowmelt timing, to warming. We even get some time travel in here. So let's go straight into the first of our speakers.
Hi everyone, I'm Emma and I'm a postdoc at UWA and Kings Park. The title of my talk today is Resting Metabolic Rates in Seeds, Relationships with Mass, Phylogeny and Climate. So seeds are living organisms that undergo aerobic metabolic processes when water and oxygen are available. And one way to calculate the metabolic rate of seeds is to measure either how much oxygen they consume or the amount of carbon dioxide they produce on a mass basis over time. Now, we have previously shown that measurements of metabolic rate measured from CO2 production correlate quite nicely with seed viability. Additionally, we have also shown that oxygen consumption similarly correlate with seed viability and also shows some promise in being able to detect viability decline in aged seeds. However, studies of metabolic rates in seeds are relatively new, and there was a need for us to understand the relationship between seed mass and metabolic rate. So in animals and whole plants, metabolic rate forms this predictable relationship with mass, which is best described by this power function on the left here. But if we convert that across onto a set of axes that have been logged, so mass here on the x-axis and metabolic rate here on the y-axis, we can see that we now have a straight line relationship between mass and metabolic rate, which is consistent across all organisms, including whole plants. For most organisms, that value of the slope, or the scaling factor, sits at a value somewhere between 0.65 and 1. So in our study, firstly, we were interested to know how resting metabolic rates in seeds scaled with mass and whether that scaling is similar to what has been observed for other organisms. And secondly, once the influence of mass has been considered, there's often some really interesting and meaningful ecological patterns left within the relationship. So, for example, are there any phylogenetic patterns present in that we might expect seeds that are more closely related to each other might have more similar mass-specific metabolic rates compared to dissimilar species? Alternatively, in the animal literature, we find that mammals from more arid environments have lower metabolic rates compared with species from more mesic locations. So we also wanted to know whether this was the case for seeds. So further, for our study, we collected data on the metabolic rate and mass of seeds from 108 different species. So we had 87 wild collected native species, 7 weeds, and 14 domesticated species. Seeds were then hydrated to 95% RH, and we measured their oxygen consumption, which was then used to calculate resting metabolic rates. So in total, we measured the resting metabolic rates from seeds spanning 4 orders of magnitude in terms of mass, and we found there to be a significant relationship between mass and metabolic rate using ordinary least squares regression. Additionally, by using phylogenetic least squares regression to account for phylogeny and making a temperature correction to account for measurement temperature, and a combination of these different analyses, we found a really similar pattern in the relationship, irrespective of the type of analysis used, and the slope values ranged between 0.75 to 0.81, which is well within the range that we had hypothesized. However, there was still a significant amount of variation within the relationship, which you can see here in the plot, by how scattered the different points are along that slope line. So next we started looking for patterns at the family level. So this figure here on the left is simply the phylogenetic tree of our study species, and then each of the nodes of each individual species has been coloured in red to represent higher than expected metabolic rates once mass has been accounted for, or in blue, lower than expected metabolic rates. And a couple of the families in here stood out as being a little bit different to the others. So you can see up here the Cyperaceae, which included 2 species native to WA, had much lower than expected metabolic rates. And then the group of Brassicaceae down here in the bottom in red all had much higher than expected metabolic rates than their mass would usually predict. So thinking a little bit further about the Brassicaceae family, the majority of species that we used in the Brassicaceae were domesticated. So we were then interested to see if any of the other domesticated species had similarly higher than expected metabolic rates. So in the figure, if I highlight the species that are domesticated, you can see that the crops do tend to produce higher than expected resting metabolic rates, even once we've accounted for phylogeny, mass, and measurement temperature. So one reason that we hypothesize might be driving this difference is that domesticated species have been selectively bred for many years in order to produce traits desirable for farming. So this includes really fast developmental rates, fast germination rates, and uniformity between the individuals, and a high metabolic rate may correlate with some of these traits. And finally, we were interested in whether the climate of seed collection location played any role in determining resting metabolic rate. So we could only do this analysis for our 87 wild native species, as they were the only species that we had really good, reliable collection information for. And you can see here on the map on the right, we've got our collection points noted in black circles. We then modelled the relationship between mass and metabolic rate, along with a series of climatic factors, including climate classification, and then 15 biologically relevant bioclim variables. So what we found was that predominantly most of our study species came from 2 different climatic regions within WA. So we had a group of species that had been collected in the southwest corner of the state, which has a much more mild Mediterranean climate, and then a group of species collected from the Pilbara region, so much more arid, much hotter, and less consistent rainfall. In the overall model with the climate variables, we found that 8 of the 15 bioclim factors to be significant, but without going into too much detail for reasons of time, each of the different factors showed that seeds from the hotter, more arid, summer-driven rainfall in the Pilbara tended to have higher metabolic rates compared with seeds from the more mesic and cooler southwest. So to sum up, we found that seed resting metabolic rate is indeed influenced by seed mass, and that the slope of the relationship is well within the range of expected slope values as suggested by other organisms, and we had a slope value of 0.75 to 0.81. We found that phylogeny does account for some of the variation in the model, but there are other factors that do play a much bigger role than phylogeny. So particularly domesticated seeds have much higher metabolic rates in comparison with wild seeds, and climate matters. And to take a saying from my colleague Dave Merritt, seeds bet their life on germination, and in the arid zone where rainfall is more transient, seeds germinate much more quickly in order to take advantage of that short window of favourable conditions. So therefore having a higher metabolic rate may therefore confer a competitive advantage in that environment. And finally, we can now correct for mass, phylogeny, etc., and use measurements of resting metabolic rates for individual species of seeds. And our current research is putting these findings into practice in order to determine if metabolic rates can be used to predict viability decline in aged seeds. So finally, a quick thank you to my collaborators on the project, and I'd like to acknowledge the support we received from an Australian Research Council linkage grant. Any questions?
G'day. So I'm Nick Schulz, and today I'm talking to you about germination strategies in an arid zone with unpredictable rainfall. So I'll be presenting the work of Dr. Corinne Duncan, who from her PhD. So our co-authors here all are indebted to Corinne and the great work she put into her PhD. Sorry. So I wish to acknowledge the traditional custodians of the lands where this research took place on Barkindji country. Pay my respects to elders past and present. So what do we know about plant recruitment in arid zones? So water availability drives population dynamics. We know that plant recruitment is typically episodic in response to rainfall, and we think that the predictability of rainfall can be mediated by can mediate germination strategies. So things like temperature and moisture thresholds and dormancy are influenced by the climate in which the plants are adapted. And we also know that understanding germination thresholds is vital for restoration and conservation efforts. And that's certainly what inspired our interest in this topic. So we were working in and around ginkgo and snapper mines in Western New South Wales, about 100 kilometres north of Mildura. And this is one of the restoration efforts underway. So the remnant vegetation includes these beautiful Balar rosewood woodlands and pearl blue bush shrublands. So seed germination usually occurs during the season of highest water availability, and optimal germination temperatures generally coincide with this period. But what about when there is no predictable rainfall season? And that's what we're dealing with here. So this is our study region here in Pooncarie with no predictable rainfall season, and compare that to an arid zone that has a very distinct wet season and a dry season. So we're dealing with something very different here. So these are our study species. We had 2 trees, Casuarina pauper and Hakea leucoptera, and 3 shrubs, Atriplex rhodioides, Mariana pyramidata, and Mariana setifolia. And the seed was collected from within a 100-kilometre radius of the centre of the study region, and all species were non-dormant except Atriplex rhodioides, and this was alleviated through an after-ripening period before the study started. And all seed covering structures were removed, like bracts. So seeds were incubated under 8 temperature regimes from 5 to 40 degrees Celsius at 5-degree intervals. And in this study, we're presenting the results from 3 water potential treatments, saturated, -0.5 and -0.75 megapascals. And we used 3 replicates of 30 seeds per species. So I encourage you to read the results of this study and the methods in more detail, and we'll just be presenting a small snippet of our results today. So here are the figures showing the germination achieved for each species under different water and temperature regimes. The wettest is always on top. So for all species, germination decreases with moisture, but we have grouped the species into 2 strategies here. Up the top here, we have our risk avoiders, all right? We've called them that because they have severely reduced germination for any combination of hot or dry conditions, okay? And these species send a clear message that when they're experiencing these conditions here, it's a no. They don't want to go, all right? Down the bottom here, we have the risk takers, all right? They have broad ranges for germination, right up to 35 degrees Celsius, even when it's not that wet, okay? So these species get a whiff of moisture and they say, let's do it, let's go, we'll take the risk. So let's think about these strategies. The risk takers wager most of their seed at each seeding event, which is fine because they seed reliably and regularly. They produce lots of seeds. They're long-lived, all right? And they also seem to be the most abundant species in the landscape. So at least at some point in time, this was a very successful strategy to use, all right? So is this risk taker strategy aided by better survival of seedlings in dry conditions? So we have some evidence of this from experiments we have conducted with tube stock planting that show that Casuarina pauper here, one of our risk takers, shows pretty acceptable survival rates with no irrigation, whereas for Hakea leucoptera, one of our risk avoiders, only the irrigated treatments had any survival, okay? So yes, maybe the ability of the seedlings to establish in adverse conditions plays into the germination strategies. But we also think that the unpredictable rainfall makes the risk-taking strategy pay off, okay? So these species have some seed on a hair trigger, ready to take advantage of climate events that are even mildly cool or wet, because these events could happen at any time. So onto our risk avoiders. All right, they are clearly looking for some combination of wet and cool conditions. They might be arid zone plants as adults, but as babies, they want a temporary temperate climate. So unlike the risk takers, okay, these species flower less regularly. They can't be as footloose with their seed, okay, which is fine, as they may only require sort of 2 or 4 large recruitment events per century to sustain their populations, all right? So they spend most of their time in a paranoid state like this, not wanting to germinate, but when they're given the chance, they will do their thing. All right, so what does all this mean for restoration? The risk avoiding strategy might be ecologically acceptable, all right, but it does not always lend itself well to direct seeding. All right, so it can be logistically difficult to coincide with suitable climate, especially if you're waiting on a rare climate event. All right, so it comes down to some species-specific decisions. All right, we need to ask for which species can direct seeding success be improved by seeding in cool temperatures. All right, and that's a possibility. And if they do germinate better in cooler temperatures, then maybe some of the direct seeding we're back on for. And for some species, tube stock planting with irrigation might be the most viable pathway to re-establishing some of these species in the broader landscape. So thanks to Ian Sluter for seed collection and guidance. Thanks to Tronox for funding the PhD project. Thanks to all my co-authors, particularly Corinne, and the work she put into this. Thanks to the conference. And thanks to everyone for listening.
Hello and welcome to my talk. My name is Sergei Rosbakh. I'm based at the University of Regensburg in Germany. And today in my presentation, I would like to talk about how snow melt timing can shape seed germination patterns in North Caucasian alpine plants. This study is a cooperation with Eduardo Fernandez Pascual from Spain, Andrea Mondoni from Italy, and Vladimir Onipchenko from Russia. So the credit also goes to them. As many of you already know, we actually live on a mountain, on a mountainous planet with a rough terrain, resulting in an extremely species-rich alpine flora. However, when you take a look at existing literature, you'll find out quite quickly that we lack any information on alpine plant regeneration by seed. For example, this famous book, the most comprehensive reference on alpine plant functioning, you'll find just a very few pages on seed dormancy and germination patterns of alpine species. This information is extremely important, especially now when the alpine ecosystems are being threatened by the continuously rising temperatures. We know that the lowland species extend their distributional ranges into uplands, thereby pushing the alpine species towards their edges of distribution. Last year, we tried to close this gap by pulling together a meta-analysis on seed germination spectrum of alpine plants. And what we know now is that an average alpine species has dormant seeds. It prefers higher temperatures for germination. Seeds of alpine species better germinate in vegetation gaps, meaning that we better respond to light and fluctuating temperatures, and we germinate quite slowly. However, in every diagram presented here on the slide, you can see that there is always some range of variation, suggesting that the local environmental factors can also affect this alpine germination syndrome. Snowmelt gradient or microtopography belongs to those local factors. And for example, in our study system in North Caucasus, here's a map for those who don't know where it is. You can see that depending on slope orientation and tilt, there might be 4 different plant communities. For example, here on the top of the hill, on the windward slopes, strong winds, you barely have snow cover, so resulting in a comparatively low vegetation period, up to 5.5 months. And here snow beds with gentle snow or depressions, where snow accumulation is tremendous, above 4 meters and higher. They develop so-called snowbed communities, and because of the high amount of snow, the vegetation period here is limited to 1-2 months only. Just once again, how those communities look in summer, you can see that visually they're completely different. And as for soil temperature and water conditions, which are important for germination of occurring there plants, you can also see clear differences. For example, alpine heath which are developed here on the tops of this hill. There's always negative temperatures in winter, meaning that the soil here gets frozen, but the main differences you can see in spring. So the spring, the botanical spring starts in alpine heat quite early because there is the snow, whereas in the snow bed at the other side of this gradient, the snow melts quite late in summer, end of July usually, and you have an increase, a drop rise in temperatures from 0 to 10 degrees. So in this study, we suggested that because of the contrasting differences in soil temperatures and water potentials, there should be also different requirements for germination in the corresponding species. So we brought the nature to the lab. So basically we used two different environmental gradients in our study, temperature gradient and water potential gradient. For temperatures, we had 6 steps from 10 to 30 degrees, and water gradient was from 0 to minus 0.8 megapascal. So basically for every temperature, we had 5 water potentials, and for every water potential, we had 6 temperatures. And here you can see that we used 18 species occurring in those communities. I will jump straight to the results. When you take a look at seed germination patterns at the species level, just for orientation, at the x-axis you have different temperatures, and the y-axis you have water potentials. You can see that there are some strict, considerable differences in seed germination patterns. Some species like Trifolium polyphyllum down here, the brighter color, the higher the seed germination percentage is. You can see that seeds of these species, Trifolium polyphyllum, can germinate basically under almost all experimental treatments, whereas seeds of some species like Potentilla verna have a very specific seed germination niche. So in this particular case, we germinate under comparatively high temperatures and low water potentials. However, when you analyze the data at the community level, you can see that seed germination patterns in those 4 communities differ from each other. Particularly, the first finding was that the species did not differ in their requirement for water. This would be this graph on the right hand. You can see that the slopes here do not differ from each other. However, we found out significant and considerable differences in temperature requirements for germination. In the next slides, I'm going to break it down for you. So you can see that as for water potential, in almost all the cases, the germination, high percentages of the germination can be found down to 0.8 megapascal, whereas temperatures requirements differ among the different communities. And of course, if you remember the differences in temperature conditions, we see that plant species occurring in those communities were simply adapted to different temperature conditions. For example, in the alpine heat, with almost no snow cover, seeds are able to germinate as long as the temperatures are above 0 degrees, whereas the snow bed communities can only germinate when the snow cover is gone, because if you start germinating at low temperatures, at zeros, it might take a few weeks or even months until the seedlings will see the sunlight. Of course, those findings, simple findings, have some important consequences. First of all, we can see that microtopography, the snow accumulation patterns, can modify large-scale alpine seed germination patterns. We also can conclude from those results that seed germination traits play a very important role in community assembly process. So basically, because of the very high specific requirements for temperature, the species of different communities know their location along the gradient. And of course, we know that the snowpack thickness and duration will be affected by the ongoing climate change, by rising temperatures. And of course, it will have some severe consequences for the alpine communities via their direct impacts. Thank you for your attention. Stay in touch. We are working hard on a paper which will appear probably in a few months.
Hello everyone, today I'm going to be talking about stepping up to the thermogradient plate. Seed germination is a critical and sensitive stage of a plant's life cycle, and climate is a key driver of starting the germination process. For example, a seed experiencing temperatures of 15 in the day and 5 at night might be perfectly viable but not germinate. However, after a change in season, or if that seed was in a different location, the same seed may be able to germinate with temperatures of 25 and 15. This kind of information can be unlocked with a traditional incubator-based experiment, but germination to different degrees is also possible at any point in between those two temperatures, and that is where a thermogradient plate comes in handy. Essentially, the thermogradient plate consists of an aluminium plate which is cooled along one edge and heated along the other. The plate is typically divided into even cells where temperature differences are roughly consistent between cells. In more modern machines, the heating and cooling edge can be switched to the perpendicular edge at a given time period to create day and night cycles. This means that each cell will experience a different day and night temperature combination. For studies that do simulate these day and night temperatures, only half the plate is usually used, as night temperature shouldn't be higher than day temperature. There are a few problems with the thermogradient plate machine that make them quite challenging to use. Firstly, they're expensive. Secondly, they take up quite a bit of space and power. And thirdly, and probably the largest barrier, is that it's hard to design an experiment with replication that can be analysed for meaningful results. Here, using 3 case study species, Electrion subdentatus from the northwest slopes of New South Wales, Colatrus baileyii from northern New South Wales and southern Queensland, and the famous Wollemi pine, we provide a guide on how to set up and then analyse thermogradient plate data in a meaningful way. The first part of our guide focuses on how to experimentally set up the thermogradient plate. We ran a number of experiments that were designed so we could find the optimal setup. These experiments included testing ways of measuring temperature, the effect of agar on temperature, and options for replication. We found that the programmed temperature can differ from the actual temperature, so you need to check the temperature of each cell throughout an experiment. You can do this with an infrared thermometer, but you need to check temperature a few hours after the transition time between day and night. Agar has a large impact on temperature, so you need to keep the level of agar consistent in each dish and measure the surface of the agar rather than the surface of the plate. Stacking dishes impacts the temperature greatly, and the use of copper tape didn't help with this. We recommend that you set up the thermogradient plate with a single petri dish in each cell, with consistent levels of agar in each dish. For example, this plate is set up with a single layer of 90mm petri dishes. In each dish, we recommend using at least 10 seeds, although you can potentially use less seeds with accuracy penalties. With no good options for replication, we use the seed itself as a unit of replication and turn to statistics to get meaningful results. We created an R notebook script that is freely available on the internet that guides users on how to plot, analyze, and predict thermogradient germination data in a highly accessible format. I'll run through the outputs of the script with our 3 case study species, all of which show a different response to the thermogradient plate. The first step is to calculate germination for each cell. To do this, we use the R package germination metrics. However, this package requires even intervals of germination checks, which is rare in the real world. So to make this package work, we created a script that automatically fills out the days that weren't checked by interpolating data between the checked days using a generalized additive model. Once this has been done, germination metrics will work and calculates over 40 germination indices for each cell, all of which can later be modeled. The 2 main indices that we tend to use are final germination converted to a proportion and the rate of germination expressed as T50. Then we plot the index that we are interested in with a rasterized quilt plot. On these plots, we put day temperature on the x-axis, night on the y, and the index of choice on the z. You'll notice that they look like triangles, and that's because the top left section is that unused section of the thermogradient plate. In this case, we're looking at final proportion germination. In our 3 examples here, we have Electrion subdentatus, which looks like it's preferring warmer temperatures, Colatrus baileyii, which looks like it's preferring cooler temperatures, and the famous Wollemi pine, which doesn't really have a temperature preference and also has low germination overall. Next, we model the data. We use generalized additive modeling, which we've implemented in an extremely user-friendly way. It's almost an automated process where the user only has to change the variable that is modeled and the type of model that is used based on that variable. Running this script will spit out this table that lists the models with a few key indicators of model fit. which I'll explain in the next slide. It will also show you the models that didn't work, and this can happen when that model isn't right for your particular data. These are those indicators of model fit. R squared is the correlation between the observed and model data. RMSE, or root mean squared error, is a common measure of model error, and mean error is a non-parametric measure calculated by holding out 10% of the data and comparing that to the model data for at least 100 iterations. Once the best model is chosen, we create those same quilt plots with our model data rather than the real data to see just how good the fit is. You can see that the model data closely resembles the real data, especially for the Electron and Collatris, which have R squared values of above 96%. The Wollemi pine model doesn't quite fit as well, and generally that is the case when there isn't a strong response to temperature. Now that we have modeled the germination response to temperature, we can have a look at real-world temperature scenarios to easily pick up patterns in germination. To do this, the user inputs a latitude and longitude from anywhere on the globe, and the script will download monthly minimum and maximum temperatures for that location. In these examples, we have inputted the location of the wild populations. We then use this temperature data with our model to predict the germination response in each month. Doing this gives a very clear indication of what the study species might be doing in the field. For example, it's clear that Electron is a summer germinator, Collatris is a winter germinator, and the Wollemi pine doesn't really care, but is more variable in winter. We can then take this a step further by downloading climate change data. In this example, the script is using the latest CMIP6 models in the shared socio-economic pathway of 585, which is sort of a worst case scenario for the years 2081 to 2100. However, this is customizable by the user. By doing this, we can see shifts that may occur in the future. For example, Electron looks like it will shift from a summer germinator to more of a spring, autumn and winter germinator. Collatris is becoming even more restricted to winter, and the Wollemi pine is again unaffected, but does see a slight drop off in the summer months. You need to be careful when setting up an experiment on the thermogradient plate. There are a range of considerations you need to make when measuring temperatures. It's our hope that releasing this script in the open source platform of R will make this analysis very accessible to everyone, including people that aren't stats experts. The thermogradient plate is a powerful tool for research, and we hope to see its use grow in the future. Thanks everyone for listening to my talk.
Hi everyone, thanks for coming to my talk. My name is Suze Everingham and I am a postdoc at the University of Bern in Switzerland, where I'm zooming in from today. But before that, I was a PhD researcher at the University of New South Wales in Australia, and in conjunction with the Australian Plant Bank at the Australian Botanic Gardens in Mount Annan. I'm also on the scientific committee for this conference, ASSC 2021, and I'm also organising an early to middle career researchers event on Tuesday evening. So if you fall in that career category, come along on Tuesday evening on Gathertown, bring a drink, and we'll have a really nice social time getting to know one another. Now onto my research. These are just a few of the images that probably come to your mind when you hear the words time travel. But I want to tell you today that there's a new way to time travel, and that's using seeds. And these time traveling seeds might help us unlock really important information about climate change, how it's impacting our plant species, and how they might be adapting to this. The way that we can use seeds to travel back in time is through this newly developing method called resurrection ecology. And that's where you revive long dormant organisms by the hatching or growing of their dormant life stage. This has been done a lot before using insect eggs and larvae, but it's also been done using historic seeds. There's a great special issue in the journal Evolutionary Applications that has a set of articles all about this newly developing method of resurrection ecology. And there's even some great examples of researchers using historic seeds, resurrecting them and testing some interesting ideas in ecology and evolution. So I was able to resurrect historic seeds from collections at the Plant Bank at the Australian Botanic Gardens and also the Seed Bank at the Australian National Botanic Garden in Canberra. I then went on fieldwork in my PhD to collect modern seeds from the same species in the same location and at the same time of year. I collected these historic and modern seed pairs from a broad range of regions all across Australia. And the reason why I collected these from such a broad area is because of the new method that myself and my co-authors have developed in this project. And this method is based off the fact that climate has not changed uniformly across Australia in the past few decades. As you can see from these figures, both temperature and precipitation have increased and decreased to varying extents across the continent of Australia in the last 30 to 40 years. So rather than just saying there's changes between the historic seeds and the modern seeds through time, we actually hypothesized that in regions where the climate has changed more, you would see greater changes in the traits of the species. And we coined this term climate contrast resurrection ecology. You can read all about it in our new article out in Ecology. So once we had collected these historic and these modern seeds, I then measured several different traits on the species, and that included their seed mass, seed viability, the seed dormancy, seed shape, and the germination rate. And those seeds were germinated and measured for their germination rate in incubators, controlled side by side the historic and the modern populations. And what was really exciting at this point was that some of the really old populations from decades ago, all the way back to the 1960s, were still showing germination, and sometimes even at the same rate as the modern plants. So the first question that we wanted to know was, have the traits responded through time at all? Were there actually any differences between the historic seeds and the modern seeds? And the answer was yes. And apologies, but from this very messy graph, you can see that the green points, or the darker points in this image, are the points where there is a significant difference within a species between the historic and the modern trait. And the orange points, or the lighter shaded points, are no significant difference. So there's a lot of green and a lot of dark and a lot of difference between these historic and modern species, except for in seed dormancy, which was really interesting, where there wasn't a lot of significant differences between the historic and the modern seeds. But the big question was, are these traits actually responding to climate change? And the answer is yes, some of them are. For example, as temperature viability increased through time, seed viability also increased. And this was found in a similar result in germination success increasing as well as temperature viability increased. And this may be due to, as the climate becomes more unpredictable and temperatures become more unstable, there might be more investment in offspring for survival and an increase in seed viability and germination success. And another result that we found was that as maximum dry spells increased, seed shape also increased. And this meant that seeds were actually becoming flatter and thinner rather than rounder, as increases in maximum dry spell happened. And that's a really interesting result. And there's not a lot of research out there on why seed shape is different within a species and what it's controlled by. So if you have any answers that you think might be occurring in this relationship, I'd love to hear them. Not only was I able to test for changes and responses in the seed traits and how they are responding to climate change, I was also able to then take these germinated seeds and grow them onto seedlings for 6 months in the UNSW glasshouse. I then was able to measure a bunch of different traits, and I found that some of the plant traits as well were responding to climate change. For example, as maximum heat wave duration increased, plant height decreased. So as it became extremely hotter and had those increases in extreme heat events, we actually saw species getting shorter in their height. I also found that as mean temperature increased, the leaf area of my species decreased, and also the leaf margin of the species became less complex or lobed, and it became more complete. One really interesting result that I found was that plant chemical defense levels actually showed no responses to changes in climate through time at all. And this might be due to indirect and direct effects of climate change on both the plants and also the herbivores that are eating the plants, cancelling each other out and showing no net difference in plant chemical defense levels through time. So some of the future directions that I'd like to follow on with using this method is taking multiple populations of the historic and the modern seeds and testing the same things. I'd also like to do successive seed collections at multiple time points and look at a more graduated change through time in the plant traits and the seed traits. And finally, I think it would be really cool to look at some reciprocal transplant experiments. So taking the modern seeds and the historic seeds and putting them in the opposite climate under controlled experimental conditions, and also keeping them in their current climate or the climate that they are used to as a control, and then potentially even taking both of these seeds and putting them into their future climate conditions. And then we can really work out what the actual adaptive potential of these trait changes are under these conditions. So my PhD research showed some really novel ways that plants are showing ecological responses to climate change in their traits. And this is really great news. But my results also highlighted the importance of ex situ seed banks. We're going to hear a range of different talks this week about the importance of managing these collections and having these seed banks to store our seeds, to protect our species and manage their risks to future endangerment and even extinction. But I think seed banks are also really important for fundamental research. And I think they're in the future going to really help us answer these novel questions about the ecology and evolution of our species. Just another reason why we should invest into them in the future. So thanks so much for coming to my talk. A big thanks to all the people that supported me, all the organisations that I worked at, to all my volunteers and research assistants, to my wonderful labs, the Big Ecology Lab at UNSW and the Plant Bank team. I miss you all so much, but it's so great to be able to see you in this virtual online platform. Thanks.
Thanks, everyone, and thanks everyone for joining the Q&A session now. Sergey is the only person who's not here, so anyone who has questions for Sergey, please pop those onto the hub, and he can answer them later in. We might start with a question for Emma. Amelia Martin has asked about why the Cyperaceae are lower than expected in their metabolic rates, and I might just expand on that more generally. With the exception of say, the difference between C3 and C4 plants, I can't imagine there being a really strong phylogenetic signal, right? So it's really remarkable to me that you're finding a big phylogenetic signal in resting metabolic rate. So for Cyperaceae in particular, but just in general, why a phylogenetic signal?
Yeah, good question. Can everyone hear me, I assume?
Yep.
Awesome. So one thing I didn't get a chance to touch on this morning was the influence of embryo size on metabolic rates. So the Cyperaceae have a relatively small embryo compared to the rest of the seed mass. So we suspect that embryo mass, being the most functionally living component of the seed as opposed to the storage reserve, is probably what's doing the most respiration within the seed itself. And that's definitely contributing to its overall metabolic rate. So it's just for a matter of time, I didn't get that into the presentation today. And we certainly saw things like the acacias, where they have the entire embryo takes up pretty much the entire seed, apart from the seed coat, do have much higher than expected metabolic rates once you take mass into consideration. So there's probably some relationship there. Phylogenetically, related species have very similar embryo size. So I suspect that's what's going on in combination with things like the domestication and impact of climate.
Thanks, Emma. I might move on to Nick. Nick, we had a question for you from Ella, and her question was about seed number per propagule. And I might just expand that one slightly. You mentioned that your risk takers were more abundant and had lots of seed. So can you sort of expand on that? Are you talking at the fruit level, at the plant level, at the lifetime level? What do you think is the driving factor there?
Yeah, for sure. So I think that there's a suite of species that all seem to fall, and admittedly we only looked at 5 species and we'd love to look at more, but there seems to be a suite of species that sort of make sense for them to fall in the risk taker or the risk avoiding strategy. And I saw the question, like Casuarina produces lots of seeds regularly, so it can burn seed whenever it likes. Okay, it can be risky, whereas the other example Ella provided was Hakea, which has less seeds, less seeding events, and I didn't present this data, but it also remains viable for longer and can experience some pretty extreme conditions. Okay, so all those traits just sort of seem to be falling on one side of the coin or the other for which strategy they're adopting in that environment. And we've got a mix of those two strategies coexisting in the remnant vegetation. Yeah.
Thanks, Nick. It'll be interesting to see as you expand the set, those patterns persist.
Yeah, for sure.
Sue's question for you from Lucy Commander. She's asking if you've looked at correlations with longevity or generation time, i.e. are there differences between seeds from trees that are 40 years old versus annuals.
Yeah, thanks for your kind comment, Lucy, and also for the question. Yeah, we were able to analyse this kind of question that you're asking a little bit by looking at plant growth form. So we didn't have a lot of perennials, so we couldn't do an analysis of perennials versus annuals, but we were able to look at growth form. So some were herbaceous, some were shrubs, and then we also had, yeah, those 40-year-old plus trees, the big eucalypts in the study as well. And we found that there actually wasn't a significant difference in the amount of trait changes that we saw between the historic and the modern plants, yeah, depending on what growth form they were. So the species were able to change no matter whether they were a really long-lived species or a really fast reproducing short generation time species. And we think that yeah, maybe these changes are occurring in different ways in different plants with different life cycles. So say they're occurring by reproductive genetic evolution in those really fast-lived species, but they could also occur within a branch, within a tree itself, that's producing those seeds through things like somatic mutations as well. But yeah, we did look into that a bit, and it would be great to get more perennials so that we could do an analysis of perennials versus annuals as well. But I could only really access the seeds that were available in the historic collections, and a lot of them are those long-lived trees. And just as I finish this question, I just want to do a second shameless plug for the Early to Middle Career Researchers event, which will be tonight at 6 p.m. on GatherTown. And if you want to access GatherTown during the conference, the password is germination with a capital G. So see you tonight if you're an early to middle career researcher as well. Cool. Thanks.
Thanks, Suze, for the shameless plug. One question for Justin. And that was, do you think that the interpolation method that you've developed would work against other germination experiments besides the TGP?
Yeah, it definitely could. To get the germination metrics package working, we've actually used that on some other studies that went on the thermogradient plate, and it worked really well. So yeah, if you were interested in doing that, if you were to download the script, the first section of the script is just that interpolation part, and that can be adapted for any other experiment.
Awesome. Thanks so much, Justin. And thanks all of our speakers for a really fantastic opening session for the conference. We will move now to a brief break. We've shoved back to the schedule by 5 minutes today to technical glitches as we get into the system. So please come back at 11:05 for our next session. Thanks, everyone.
Thanks, Adrienne.